taught this a little more in depth. When you have a sentence like this, right, if we have a 3-letter codes in a sense, right, 3-letter words, and, you know, we change one of them, what's another word? Bad cat, yeah. Right, maybe that one make that much difference in a sentence. It changes it. Maybe it's a better sentence, maybe it's a worse sentence, but it's still a sentence. But you start to take a letter out, right, or add a letter, well now everything's off and it doesn't make sense at all anymore. Well, we got a her in there, but yeah. All right, down here then, sickle cell, nothing really different there. However, you're likely to see, I'm planning to give you a little bit more information about the sickle cell and have you guys try to figure out actually what happens, why sickle cell, why that one change makes a difference, I guess. So that is the major difference. I believe hemoglobin has something like 250 amino acids in each of the 4 chains. So this is one amino acid out of all those, and well, I've had it twice. I've had it twice probably, but so 2 out of about 1,000 or so changes, and makes for a difficult situation for people because they don't tend to live very long, at least in the old days. Okay, you okay with that? Mutations? So, I mean, that's all I'm going to say about it. I didn't really cover that in the notes, of course, anywhere. But you'll see some examples of that. I'll ask a couple questions like that in a few days. I'm still working pretty hard to try to come up with exactly what you guys are going to do. I mean, I have a pretty good idea, but most of that stuff is made. Some of it is just things I've done before, but some of this new stuff, we're covering new stuff, really. I haven't really taught before. That. So let's go back. We had some notes that we were working on. I think I showed you that again, didn't I? That video, Human Genome Project video. That's a good one to watch if you're having trouble. Started this, I believe. Yeah, we did. We're going to start from the beginning on this, I think, again. So these are the 2 ways, 2 locations or time frames. We have changes in transcription, we have changes in translation. There's part of the notes that you're filling in there.
So the first method is DNA passing into chromosomes. When you take a look at a
Shaliba, I caught this kid trying to skip class. So.
Oh, good.
Yeah, that was
Thank you,
close.
Mr. Holmes. He usually gets away with it.
When you get DNA to sit nice and neatly inside the nucleus, this DNA is going to wind around histone protein, like this, and it looks like the enzyme. The winding of DNA around the histone protein forms the structure called a nucleus. The nucleosomes then line into a bigger helix. And then that helix lines into a supercoil. And then the supercoil moves and folds. And that's actually what makes up a chromosome. So a simple picture of a chromosome may lead you to think that the structure is pretty simple, but there's multiple levels of DNA paths. And this is important to transcription because in order for RNA polymerase to transcribe DNA, the DNA has got to be unpacked. If the DNA is packed up like this, RNA polymerase can't access the gene and it can't transcribe the DNA into an RNA.
I don't recall, I didn't preview this again this morning, so I can't probably say more about this, but, so RNA polymerase, just here at the bottom of your notes, what is that, what's that do? Ends in ACE, it's an enzyme, pardon? It's an enzyme?
Yeah.
What's it do in this case? Polymerizes, yes, the RNA, so it's making the mRNA, okay? It actually opens up the DNA, because the DNA has to be separated to make our proteins, right? We have a code somewhere on one of these strands for protein, and to make the mRNA, it has to open up. So the RNA polymerase does that. What they're saying here is, if this doesn't uncoil, the RNA polymerase will never work. Right, so that's one transcription factor that might, yeah.
What does helicase do? It's not on there.
Helicase unwinds it, yeah.
Is it-
breaks it
Oh,
up.
yeah, I'm sorry, yeah, so the
So-
helicase actually unwinds it,
Yeah.
and then the RNA
Oh, okay.
polymerase
It separates the
works
nucleotides.
to initiate the,
Okay.
Isn't it the one that also puts on the nucleotide bases,
pardon?
Doesn't it also add the nucleotide bases?
or the RNA bases?
Yeah.
Say it again?
Does polymerase also add the nucleotide bases? Like-
Yeah, I think it assembles the
Yeah.
nucleotides, yeah. Yeah,
Another
so,
method of transcriptional
so the RNA
regulation is something
polymerase
we're
can't
pretty
work
familiar
if we
with,
can't get
transcription
this
factors.
unraveled.
Here we've got a DNA strand. The gene is right here. This is the part of the DNA that will actually code for a protein. Before the gene is a sequence of DNA called the promoter. We know that the RNA polymerase has to bind to the promoter in order to start transcription. But then, further upstream,
Okay, so just what's the role of the promoter then? Yeah, the RNA polymerase has to bond to that. Remember all, what's our DNA made out of? Sugars, phosphates, which aren't as important to us, but the bases then, right? A, C, T, and Gs. All right, so we got this promoter region, all right?
from the DNA promoter and gene, are these sequences called enhancers. Enhancers will get bound by transcription factors. And the goal of transcription factors is to help RNA polymerase start transcription. So here's what it looks like if the gene is going to get transcribed. Here we can
All right, so in this diagram, they're showing us that the enhancers are required, right, for all this to get started. I'm not sure what you're gonna say they are. Really, I usually see these listed as like a gene switch. Yeah, more like a gene switch, or just call them switches, I think, that they turn on or off, our promoter and our gene. Yeah.
Wait, so like one enhancer goes off?
Pardon?
Oh, wait, no, I see.
You'll see that, I'll go over this in the end, you're not gonna have to know a lot of this, or any of this in a way, but I want to point out what some of these other aspects are of gene regulation. And then we have the transcription factors, which seem to be other substances you can just require, okay, and the enhancers.
see that the transcription factors are bound to the enhancer region, and in doing so, they have helped RNA polymerase bind to the promoter. Now RNA polymerase can start transcribing the gene.
So part of what I'm trying to get across here is that we need some things outside the gene. The gene is where the actual protein is being coded for. all right, that has a message for the amino acids that are gonna make the protein. But there are other parts of the DNA that are important. And if we have mutations in these parts or changes in these parts, that might change how the gene's regulated. Some of them are pretty close. There's usually this one, the promoter, is generally right in front of the gene. Some of them can be kind of far or further away. Okay? And that's really what I want you to know about this. You don't really need to know these names necessarily and what these things are, but
So to sum that up, RNA polymerase is the one transcribing DNA into RNA. The promoter is the green region that the RNA polymerase has to bind, and the promoter is DNA.
I'll stop
The
this in a minute.
enhancer is another area of DNA, often quite far away from the gene, and that's what the transcription factors bind to. And the transcription factors are these purple and green markers.
All right. So, you don't copy all this down, but, we need the promoter to initiate transcription, transcription maybe is what I would say. The enhancer, often far from the gene, that's maybe more important to me. And then the transcription factors are regulatory proteins, which of course if they're proteins, then they have to be made somewhere else as well, right? They have a gene for them. And the polymerase, of course, also comes from a gene, right? All these things come from other segments of DNA. And that's the point that I really want you to make here. Well, if you feel like you need to. Yeah, yeah. Okay. So let's start
And it's
working on what
important to
these
note,
do,
if you want
what
to
they are.
add this to the transcription factors can activate or repress transcription. In this particular diagram, we are seeing activators, so they're helping activate the RNA polymerase. But there are other types that will repress RNA polymerase's activity and turn things off. Another type of control is our alternative splicing, and we're familiar with this as well. Remember that in a DNA gene, not all of the sequences will end up in the final RNA. So the DNA gets transcribed into RNA, but only the exons, 1, 2, 3, 4, and 5, end up in the final RNA molecule. The introns, these non-coding regions, get cut out and discarded. And the cool thing about splicing is that you can splice the exons together in different ways. For this mRNA molecule, we splice together 1, 2, 3, and 5 to get a particular mRNA molecule. In this one, we splice together 1, 2, 4, and 5. So the cool thing about this is there's one.
I don't think I mentioned this here class that I recall, maybe it was, but when I was in college, they thought we had one gene. Did I tell you guys this? That there was one gene for every protein. They thought there were a million proteins. And so they thought there were a million genes. Well, when the Human Genome Project came out, there were more like 20,000 genes or 30,000 genes in the human. So it was like way less than what they predicted. And they kind of thought people were doing it wrong and they made mistakes. But one of the reasons we don't need so many genes is because we can rearrange these into different proteins in the end. Okay, so we get lots of use out of these genes. So we have our introns that are cut out, or exons that are put together. I guess part of what I would want you to get here is that there may be parts of the DNA that don't really matter, right? We're going to cut them out anyhow, so like a mutation in here wouldn't really matter. Or maybe even a mutation here wouldn't matter for this particular segment, but it would for this one, right? So we have parts of the DNA that aren't as important, even though we might say it's within our gene.
We can get multiple.
Are introns genes or just like the in-between stuff like the promoters?
Anybody help me for that? Would they consider the intron part of the gene?
DNA that don't code for the protein.
Do they consider that part of the gene still?
Well, it can, for some, right, because if you, like you said, if you rearrange it, sometimes
Oh.
introns are different. for
Yeah,
the
yeah, so
exon.
sometimes the intron
Right, so
isn't an intron,
sometimes
it's
the
an exon,
introns-
huh?
yeah.
Yeah. So, I'm not sure we answered that question
So, I mean, if
very well,
it would
but,
probably be considered,
but I would probably say it's part of the gene.
So there are genes that like don't code for protein? Is that-
Yeah, so some parts may be cut out, so they would not ever be transcribed in protein.
RNAs, which means multiple proteins. And this helps explain some of the variation in phenotypes that we see within humans or with humans compared to chimpanzees. Because even if two different people have the same DNA sequence, you might get different traits depending on how the RNA gets spliced together. Another method of controlling transcription happens slightly after transcription, in fact. Here's our mRNA molecule. You can see it's
I think we don't have to worry about this one at
but
all.
eventually the mRNA
Not worried about this
molecule
one
is
at
going to
all.
get broken down or degraded. And when that happens is key. Because the lifespan of mRNA determines how much protein. It won't degrade the mRNA, right? And there's a specific mechanism involved in degrading mRNA. It's called RNA emission. For a long time, scientists just thought that 90% was done, but it turns out that it's not really done. In fact, some of it is transcribed into tiny little RNA molecules called microRNA. And here's what those microRNAs do.
You really sort of want to
A
emphasize
microRNA
here is that
is
there's
a
lots
short
of
little
other
RNA.
little places
It
where
gets
we
together with
need
some protein, and it will bind
information
to
from our
an
DNA.
mRNA molecule. And once it binds to
Essentially, we're going to
that,
stop, okay?
Like,
I'm not emphasizing so much about regulation. I want you to know a couple things. The main thing I want you to see here is there's several ways we can regulate protein production. But that mainly what I'm trying to show you is that there's other parts of the DNA that are important for producing a protein other than the gene. We've got this promoter site. We've got these various switches. So that may not make sense as to what it is I want you to know just yet, but I think it's got to come clear next time maybe, or next activity, not today really. Probably not till Thursday, actually next week. So let's see what I've got here. So don't worry too much about that part of notes. I don't recall what this one is now. Maybe I can't remember anything anymore. Put these notes together like 3 days ago. Oh, I think this is the one that's pretty poor quality video. No?
So this is a gene with the yellow part of the DNA, the coding part colored yellow. That might encode something like a lens
This is
protein.
similar to what you just
It would
saw.
normally only be expressed if an RNA polymerase lands on the gene's promoter and makes a messenger RNA from the gene. The coding region of the gene is surrounded by a series of regulatory switches. So these are parts of DNA that don't code for any protein. Instead, they act as switches that determine where and when the gene turns on. Those switches are the landing sites for regulatory molecules that bind to the switches, recruit RNA polymerase for the gene's promoter, and cause an increase in the total number of messenger RNA transcripts coming from the gene. Typically, a gene will be surrounded by multiple switches that allows the gene to be turned on at different times and places under the control of different signals and regulatory molecules. A lens protein in the mouse is also expressed in the liver, for example. It might have a switch where a regulatory molecule turns it on in the eye lens. The PAX6 gene is an example of a regulatory molecule that would bind to one of those switches. There'd be a different switch to turn the gene on in the liver.
All right, just see here some diagrams of this. I've got a coding section, got promoter, non-coding. The RNA polymerase binds to that polymerase, promoter site to initiate transcription. Write that in your notes. In many cases, we have switches in addition to the promoters, one or more switches. Sometimes these things are behind. They call us upstream from the gene. Sometimes they're downstream as well. But generally, most of these things are upstream. Remember, we're reading a bigger direction when we say that. 5 times 3 times the direction doesn't matter. Here's just a regulatory model, molecule